Good morning. I would like to welcome everyone to today's MD Anderson Cancer Center Enjoy Science seminar. This is a series of lectures given by the MD Anderson faculty members and organized by the Office of the Chief Scientific Officer, Dr. Giulio Dretta. I'm Alessandra Ferraioli and I have the pleasure of hosting this series with my colleague, Dr. George Kalin. The goal of the Enjoy Science initiative is to strengthen the connection between the scientific and medical community at the MD Anderson Cancer Center and our colleagues, our patients, and our friends around the world during this time of limited in-person, at the bench, and in the clinic activities. Today's seminar is being recorded and will be made available on the Enjoy Science website later this week. The speaker of today is Dr. Kunal Rai. Dr. Rai is an assistant professor in the Department of Genomic Medicine at the MD Anderson Cancer Center. Dr. Rai leads an active research laboratory that focuses on understanding the role played by epigenetic changes during cancer progression, and he has extensively published in this field. Today, Dr. Rai will be discussing for us the topic of the cancer epigenome, junk to gold. Dr. Rai?
Thank you, Alessandra. Can you guys hear me well?
Yes, we can.
Okay, so let's get started. It's a funny feeling giving a presentation in these times of COVID with just you feeling like you're talking to your computer without any audience. So suffice to say, there's a huge value of a human-to-human interaction while the audience is sitting in front of you and nodding or not nodding, and which makes you feel much more either appreciated or takes your pace differently based on the reaction of the audience. So hopefully I do justice to this, and there are no silent gaps in between. But it's a funny feeling giving these presentations, though. So just to justify the title of my talk, I guess I'd start with this slide which essentially in 2001, when the first human draft genome was sequenced, and it was figured that most of the genes that were around at that point, they thought it was on 30,000 genes or so. They make around 2 to 3% of the genome. So a subset of scientists thought, oh, is 97% of your genome just junk DNA? Really, but hey, just because we don't understand doesn't mean it's junk, right? As a scientist, obviously, we want to understand what's the function of this non-coding genome. And therefore, we turn to something called epigenetics, which a field that started in 1940s or 50s, where and this is one of the phenomena that actually led to the development, most of the development in epigenetics, which is called position effect variegation, for example, seen in maize on the left slide here, in Drosophila eyes, or in the coat colors in mice, or just in the colors of eyes, as you can see in this particular human being. This does give the findings were that if you have the same DNA, how is the phenotype so different? And that's what led to epigenetics. So suffice to say, there's a long history of various activities in this field, which essentially led to the state we are in today. And this is a relatively old slide, but this does tell you major discoveries made in this field over time. Excuse me. And this essentially led to the state of the field as we understand it today. And the discovery of next generation sequencing techniques obviously led to a revolution in epigenome mapping efforts as well. And these are some of the mapping efforts that have been done in last 10 years or so. The results have only come from 2012 onwards. So it's still an active field, it's still developing field. Most of these projects actually And these are multi-PI, multi-million dollar projects, which essentially have mapped epigenome in, in normal tissues and different aspects of epigenome. And I'll come to that in a minute. However, there has been no systematic efforts in cancer. So that's the focus of my lab in last, that has been the focus of my lab in last 5 years or so. So going back to the junk DNA idea, in fact, when the ENCODE, which was the first major consortium data uh, published in 2012, it said that essentially in the abstract that now we define biological functions for the 80% of the genome, which again has its own set of controversies and led to even big books being written about it. So and when we and with this sort of evolution with the genetic as well as the biochemical evolution, this is where we come to, and essentially uh, there is genetic evidence meaning essentially which parts of the genes actually drive a phenotype, and therefore then the gene where the genes are present, there is evolutionary evidence for parts of the genome, and then there is biochemical evidence which is encoded by our roadmap epigenomics focus. So essentially we now know functions of, well, at least in theory, functions of 80% of our genome, but still the rest 20% needs to be deciphered. It needs to be kept in mind as I said earlier, most of these studies have been done in normal context. So what about disease context? And epigenetics obviously is thought to play major roles in disease as well. And this is an example of a monozygotic twins where both the twins carried an F1 mutation, but one exhibited the phenotype later in life, another did not. And this is a famous case of a UK comedian, I think. So, and there is case to be made here that this particular case could be driven by epigenetic modifications or epigenetic events in during the history of the disease. In cancers, there is quite a bit of evidence, and we'll be talking about that. And first of all, just aberrant chromatin structure is a key feature of cancer cells as such. The nuclear to cytoplasmic ratio is one of the things that is used by criteria used by pathologists to decipher the cancer cells, for example. So the question we like to ask in my lab is, can we exploit or can we understand, first of all, this rest 98% of the non-coding genome, and can we then exploit it for cancer therapeutics? So what are these epigenetic elements in the cell? So as you can see here, this is essentially your interface nuclei, which with the DNA wrapped around in within this, the 2 meters of DNA is wrapped within a 10 micrometer of space in the nuclei, and uh, this is the sort of the structure where you have an higher order structure of the nuclear of the DNA in the 3D space that leads to the specific wrapping of the DNA around these nucleosomes. And then there is other elements. There's modifications of the site of the DNA bases per se. So there is 3D chromatin structure. There is nucleosome positioning, where nucleosome is positioned. So what is available for the external protein factors to access the DNA. And then there is modifications of this nucleosome, which essentially has these unstructured tails that are floating in the nucleoplasm, which make a major epigenetic element. And then finally, as I mentioned, there's modifications of the DNA base. Pardon me, I have a bit of a cough today. So what are these histone modifications? So, okay, my lab's focused a lot on the histone modifications. So what are these histone modifications? So as I mentioned, there is these unstructured tails of the nucleosomes that carry these covalent modifications, which are methylation or acetylation or phosphorylation, ubiquitination, and then there are many more now known. This is a slide from 2007 for that matter. And there are two copies of each of these histones in this nucleosome, 2H3, 2H4, 2H2A, 2H2B. And these get modified with a wide array of different modifications. In fact, over 100 such modifications are known to occur in nucleosomes. So the importance of this field in cancer actually came quite a bit to highlight um, from the studies of TCG and ICGC, which found somatic mutations in various components of this chromatin machinery in cancers. And this has become a major paradigm or hallmark actually of cancer now. So in the last 8 to 10 years, various studies the roles of these mutations in these proteins in different cancers. And how can we therapeutically use the selection of these mutations in different cancer types? So mutations can occur in methylation, DNA methylation machinery. So this is cytosine methylation machinery. It can occur in the chromat reader proteins that read these modifications. It can occur in the remodeler proteins, which actually move these nucleosomes. And it can occur in just proteins that write, which are here on the bottom, or erase these marks on the histone tails. And indeed, that led to a huge focus in the pharma industry for development of therapeutics focused on epigenetic proteins or these various molecules that are mutated in cancers. And so much so that in 2012 there was a small article in Economist regarding the focus of epigenetics, regarding focusing on epigenetics in cancer therapy. And indeed, that has led to development of inhibitors for different enzymes and chromatin proteins, which are some of them are listed here. The ones the ones in the red actually are in clinical trials or in clinic already, and there are a lot more proteins in clinical trials now. This is a figure from a 2016 review article, and suffice to say, this has actually generated huge interest, as I said earlier, in pharma company, and there is lots and lots of efforts for making clinical grade compounds for these proteins. And what is important is to utilize these compounds in specific genetic setting where either those specific mutations occur or specific epigenetic alterations occur. So that's the message number one. There is a huge body of work out there which suggests that epigenetic proteins and domains can be target of therapeutics, and you will see more and more coming up as we go forward. So I want to come to the focus of my lab. So what about the alterations of epigenome in cancer? So just for DNA methylation, which is a major prominent epigenetic mark that actually started in 1960s, there's been two types of events known. For example, there is specific tumor suppressor focused hypermethylation shown here. And this occurs on major tumor suppressors such as CDKN2A, MLH1, P10, etc. On the other hand, at the global level, there is loss of DNA methylation, which is called hypomethylation. So this is a opposite phenomena. And since a major portion of our genome is made up of these repeat regions, if you just look globally, you see a major hypomethylation in cancer. Both of these events are actually being utilized now for therapy, obviously. Moreover, DNA methylation actually tends to be specific to a subset of patients. And I'm giving you an example here of SIMP phenotype, which is a hypermethylator phenotype. So if you see here this cluster 1 in yellow, these patients can exhibit this SIMP phenotype, which actually could be target of specific therapeutics, especially focusing on DNA methylation inhibitors that could target this set of patients. It's interesting to note that in this particular disease, this SIMP phenotype actually contains mutations in BRAF allele, which is itself targetable, and is a huge target in melanoma, for example. So there are efforts underway to combine BRAF mutant and BRAF inhibitors with the inhibitors to DNA methylation to target this specific group of patients. Now coming to the chromatin elements, there are various type of regulatory modules in the cell, and some of those are listed here. For example, promoters and insulators, enhancers, and these are the major protein. These are the major elements that essentially regulate how a gene um, gets transcribed and how many copies of the gene gets transcribed eventually. And I'm talking about that because people have found also by these whole genome focused studies that there are mutations that occur either in the promoters, active promoters, so promoters get preferentially more mutated as from this Nature paper. There are mutations in the CTCF binding sites, as well as a more recent study from our colleagues in our department showing the alterations in chromatin domains by these mutations or somatic variants. So that makes the case that not only proteins are mutated, but also specific epigenetic elements are mutated. However, most of these studies utilize epigenome maps of normal cells and tissues that I told you about, either from the roadmap epigenomic study or the ENCODE study. In my lab, we like to decipher, we'd like to ask, are the epigenome maps of cancer cells different than the normal cells? And if so, then a lot of the results will change. Hopefully or not, we'll see. So I'm going to tell you about a couple of stories and just give you some glimpses of that. One of the stories focuses on our recent most profiling of 145 cancer cell lines. So these are cancer cell lines that have been utilized, that are very deeply profiled by CCLE and Sanger Institute efforts. And we focus on 9 different tumor types here. We looked at 6 different histone modifications, as they can be markers for various epigenetic elements and allow us to learn about the transcriptional states of the associated genomic loci. So for example, H3K4 mono can be a marker for enhancer, and H3K twin acetylation can be a marker for active enhancer. When they are present together on the same nucleosome, they can mark an active enhancer. 79 methylation, for example, tells you the gene expression for can be a proxy for gene expression. K4 trimethylation is present where RNA polymerase is poised to go on and transcribe. So these are activatable promoters. And then there is K9 trimethylation, which actually tells you about heterochromatin. And then K27 trimethylation that tells you about polycomb silencing. So a general approach in my labs to profile these histone modification marks using high throughput chip sequencing based methods, and then learn various chromatin states that occur in cancer cells and how they are differ from normals, and then ask if these can inform on various pieces of biology, whether it's a specific biological insights or can they be used as prognostic markers, or can they be used for specific targeted therapeutics. Sorry. So in this particular study, we profiled 6 marks, and then various other data sets were already available from the CCLE or Sanger data sets. So we utilized all of these for our studies. So the first concept that I wanted to get you here was that when we looked at the enhancers, we found that and compare them to the normals. So the normals are all here 93 different normal tissue data sets from roadmap and ENCODE. And on the right side is, in the circles, are all the cancer cell lines. What you see is, for the most part, the cancer cell lines segregate away from the triangles, meaning the normal tissue. We suggest that enhancers may be the basic fundamental features, or enhancer aberrations are the basic fundamental features of cancer tissues. Note that these do not necessarily segregate based on their tumor type. So it's not like a blue triangle is with blue circles, or a pink triangle or purple circle is with purple triangles. Okay, they're quite segregated, which essentially says that irrespective of where they come from, which tissue type they come from, they segregate away from normals together. So that's an important concept. And indeed, there have been other studies that have shown that, for example, in colon cancers, there's been showing studies showing that hyperactivation of various enhancers and so on and so forth. So there's growing literature that suggests that epigenomic abnormalities are fundamental features of cancers. So I'm talking about enhancers. So what are enhancers? So enhancers are modules that essentially have a higher density of transcription factor motifs. And transcription factor binds to these enhancers preferentially. Then they recruit mediator complex, which essentially then talks to the gene promoters. And as that mediator complex brings them together, the promoters and enhancers, so eventually activate gene transcription. There's a term called super enhancers, and super enhancers are essentially a higher density of enhancers within, let's say, a 12 kb region. That's a norm that is used. And what that is thought to do is to actually integrate multiple signals from the cell surface. So for example, if the key regulatory event uh, gets a signal from various different signaling events such as LIF, TGF-beta, and Wnt as depicted here, the transcription factors that eventually bring the signal, relay the signals of these signaling processes are built together in these enhancer, super enhancer elements. So that way, you can actually integrate the signal much easier. And the genes that seem to harbor these super enhancers in their vicinity seem to be the key regulators of cell fading, at least in the normal developmental context. In cancers, these super enhancers are thought to occur around the oncogenes to activate their gene expression to a higher level. That still needs to be established well, though. Whereas typical enhancers will normally integrate signals from one different signaling event. Now, what's interesting is that in different cancers, these enhancers or super enhancers for the same gene can occur at different genomic loci. So for example, shown here is an example of pancreatic cancer, TLL, or colorectal cancers, which show where the enhancers or super enhancers exist for the major oncogene MYC. And you can imagine if you had a therapy targeting a specific enhancer or a specific genetic element tomorrow, you may want to know in that particular patient where is the super enhancer located. So that's the significance of the work we've done in this space. And here I'm just showing you one snapshot from various different cancers. And what you see here, if you focus on the yellows are the enhancers. And this is again the MYC locus. MYC is right here. And what you see is the enhancers are located at different positions in these different cancer types. Sometimes even within a cancer type, you can see differences in where the enhancers are located. So that speaks for understanding where crucial regulatory elements are located in cancers, and then how could we then utilize them for therapeutics. This also tells us that there is a built-in heterogeneity among cancer patients. So just if you apply the idea of the personalized medicine to the epigenetics or epigenome, what you can potentially epigenome can be utilized to bucket patients into different groups. Could have blue groups with a certain epigenetic makeup, and the yellow groups and the red groups and the green. And indeed, when we compare, as I said earlier, our data to the cancer data to the roadmap data, we see various enhancers that are cancer specific. So that obviously answers first of all, that answers the question. There are quite a bit of unique set of cancer specific enhancers. And that holds true for other epigenetic elements as well. And these can be different between different cancer types, as shown here on the pie chart on the right. Only 20% of them are shared amongst all the cancers that we actually profiled. And when we went on to look for heterogeneity among cell lines, so this is only all the cell lines that we had, so 142 cell lines here, 125, I'm sorry, cell lines here, For this, we essentially used an NMF clustering based approach to cluster the samples. And what we found was there were 5 different clusters based on this enhancer subtype. Now, these different clusters are shown here on the bottom. And when we associate them with the cancer subtypes, the histological subtypes where they come from, you can see that some of them correlate very well. For example, EPIC1 is mostly a melanoma derived cluster. And that makes sense because melanoma is actually a neural crest derived tumor. However, we have another tumor called MPNST, which is also neural crest derived, but that is actually spread out among others. So that's interesting. Melanoma is a different enhancer profiles than everything else. EPIC2, which essentially is a breast cancer like breast cancer and ovarian cancer focused subtype here. We call it gynecological or hormone driven subtype. And then EPIC3 essentially harbors cancers that are colon cancer and pancreatic cancer. When we overlay it with the TCGA data, we also see stomach cancer coming. So essentially it's a GI-based subtype. So that speaks to their developmental origin, which is coming from the endoderm of the gut tube. Interestingly, we also found another cluster, EPIC4, which essentially is made up of different colors, which means irrespective of their histological subtypes, they share their enhancer features. So they are more similar in terms of their enhancer profiles rather than their histological subtypes. And finally, we also have another cluster which is also mixed, but contains majority of head and neck cancers and the GBMs. What was interesting is that none of these clusters correlate very well, at least with the known mutations or known bona fide oncogenic or tumor suppressor mutations, except that PTEN is actually PTEN deletion. So 4 out of 5 PTEN deletions are enriched in these EPIC4 clusters. So there is a hypothesis out there that somatic mutations occur and then they change the epigenetic landscape over time, and then they together build the tumorigenic phenotype. However, this data would suggest that potentially enhancer aberrations arise separately from the bona fide oncogenic mutations or tumor suppressor mutations, and then they might act together to drive the tumorigenic phenotype. Now, if you look at different EPIC phenotypes, they do correlate decently well with their RNA expression cluster. So that also tells us that enhancers are probably a more important determinant of their gene expression patterns at the end of the day. And these are just the pathway analysis for these different clusters, and you see the specificity in that, at least somewhat. And interestingly, EPIC4 has increased PI3-AKT signaling, which is a major downstream signaling affected by PTEN deletion. So that's consistent. So we focus a little bit more on EPIC4, and just to summarize, we can identify a subset of TCGA samples that are multicolored, meaning they come from all different tissue types and have EPIC4 signature on it. So, and more importantly, this EPIC4 signature tumors actually had poorer survival compared to the rest of them. So there is a need then to understand biology deeper of this particular cluster and identify their specific therapeutics in this space. And as I said earlier, there's PI3 kinase pathway activation here, which is consistent with the RPPA data that shows higher levels of AKT activity in this EPIC4. And that's also consistent with slightly better AUC values, lower IC50 values for PI3 kinase pathway and AKT inhibitors in EPIC4 compared to the rest. Oh, no, sorry, I'm sorry. This is AUC values, better AUC values in EPIC4 compared to the other clusters. So next we asked if we could potentially identify a specific core TFs. So now let me take you back to the IPS model where core TFs such as OCT4, SOX2, NANOG together can take a differentiated cells and convert into a more pluripotent or stem-like phenotype. by just overexpression of these three. A lot of work from various labs and most prominently from Rick Young's lab has shown that these three key transcription factors can do so because they self-regulate themselves as well as they regulate a bunch of other transcription factors that go on to activate major transcriptomic networks that eventually drive the phenotype. So to do that, we asked if in each of these subclusters of cancer cell lines, could we identify core driver transcription factors that can drive this phenotype. But what we also did at the same time, using this algorithm on the right, which essentially takes into account the self-regulation and the motif presence in the enhancers as well as their higher expression. But what we also took advantage of was Avana data, the CRISPR dependency data in these cell lines, where we asked for each of these clusters, once we identify the TFs that drive these, which ones of these are really, which of these clusters are really dependent on these core TFs. So essentially then allowing us to identify factors that could potentially serve as therapeutic targets. And indeed we found a bunch, few in each cases. And one of the examples from each of them is shown here. So SOX10 would be an example for EPIC1, which is melanoma focus. And then SPDF would be one for the EPIC2, CDX2 would be for EPIC3, ZEB1 would be for EPIC4, and then GLIS2 will be for EPIC5. So in summary, we have identified a few epigenetic subtypes of cancer cells, and they have unique features. They have unique histologies, and where importantly, EPIC4, which might have more mesenchymal features, might be the one that needs to be targeted further for therapeutics. And we are actively working on that area to identify what might be the major inhibitors that could be used for that particular subtype. And so first, and another message that I wanted you to take was that there are core TFs that could potentially serve as therapeutic targets in specific subgroup of patients which are defined by a peculiar enhancer signature. And indeed now undruggable is no more an undruggable thanks to new technologies such as PROTACs that actually can target any protein as long as you have a molecule that that binds with a high potency to your target protein, and you can link that with the molecule that takes it to takes the targets your protein target to the ubiquitination machine for ubiquitination by the for proteolysis, eventually for degradation. So obviously these are being actively pursued in the pharma industry, and we are working to identify molecules against core TFs for specific enhancer subgroups that we have. So next another set of questions that we've been asking in the lab is, can we use enhancer epigenomic signatures for stratifying patients either within a specific subtype or for specific therapeutic responses. So I'll tell you about the first thing first in next few slides, and then I'll come to the therapy response. So here I'll take an example of the colon cancer study. Just wanted to focus specifically on one cancer. So it's colorectal cancer. This is done with the integromics group at MD Anderson. And we had around 60 or so samples where we profiled using our favorite method, ChIP-seq for 6-histone mice, as I mentioned earlier. And at the same time, we also profiled a bunch of cell lines. Here, just using now the tumor data, what we were able to identify, and similar approach as I just showed you earlier, is the EPIC subtype for colorectal cancers. So there are 4 subtypes of colorectal cancers. So here, there is not multiple tissue types. So this gives you a more purer feeling as to what happens within a given tumor type. So what we found was, again, 4 subtypes of colorectal cancer called EPIC 1, 2, 3, 4. And what was interesting was they didn't really associate with any of the clinical data, which is on the bottom, but they did associate well with their transcriptomic subtypes. At least 3 of them did. So see, EPIC1 correlated very well with CMS1, EPIC2 correlated very well with CMS4, and uh, EPIC3 and 4 bifurcated CMS2 into two subtypes. So that has its own set of interesting connotations to it. So based on the fact that, well, okay, so let me tell you about the CMS subtypes. The CMS subtypes are essentially defined uh, transcriptomic signatures of colon cancer. They are designed based on the RNA-seq data. And essentially what you find is four subtypes again based on this transcriptomic signature, which are immune, canonical, metabolic, or mesenchymal. And as I said earlier, EPIC phenotypes correlate very well with the CMS subtypes. Indeed, if we just look at the pathways for genes around which the enhancers are gained in each of these EPIC subtypes, we see very similar pathways to ones that have been defined by the RNA expression signature. So that tells us again that enhancers are likely drivers of this transcriptomic output. And with that, we hypothesize for therapeutic purposes, can we block enhancers to lock cancer epigenome in a specific state and further hit the cell with inhibitors of specific driver pathways in each of these EPIC and CMS subtypes? So we did that using epigenetic inhibitors like bromodomain inhibitors that block enhancers, and then went with specific pathways inhibitors. And indeed, we see very specific responses in that CMS1 is actually responds much better to the PAR plus bromodomain inhibitor, whereas CMS2 and CMS4 don't. And CMS2 then responds very well to EGFR plus bromodomain inhibitor, and CMS4 responds very well to TGF-beta plus bromodomain. So there's a built-in specificity that can be achieved by using a bromodomain inhibitor along with the pathway inhibitor in these cases. So that's our take-home 4, which is essentially epigenomic content of a tumor can be potentially useful for design of novel precision therapeutics. And next, we wanted to apply the same model to immune therapy because obviously immune therapy is come to the forefront in cancer therapeutics. And indeed, there is a bunch of studies out there showing that epigenome could be a modulator for immune therapy response. So we teamed up with Jen Vargo's team and the melanoma group here, and we identified around, we obtained around 40 samples from a trial on anti-PD-1, as well as another 30 some samples on CTLA-4 trial. And we did our favorite approach again, profiling 6 histone marks in these biopsy models. And just to not undermine, this is not trivial. I mean, profiling these 6 histone marks from a biopsy is tough, but we were able to achieve that thanks to some of the wonderful technical abilities of some of the postdocs in my lab. To make a long story short, here we identified a signature of enhancers, and this is around 32,500 loci in the genome that actually gain enhancers in the pre-treatment stage, so even before even the treatment started. And they're gained in the non-responders consistently, and this is around 20,000 or so of those loci, just focused on one histone mark, K27 acetylation. So this could potentially be taken to a CLIA type of setting and be converted to a biomarker approach. As I said earlier, K27 acetylation is higher in the non-responders in the pre-treatment samples compared to responders, and the opposite mark, K27 trimethyl, is higher in a subset of these loci in the responder samples. We then collaborated with an MIT MGH team who had around 70 patients there who had profiled for XTK27 acetylation. And we were able to, at least on a subset of these peaks, we were able to reproducibly see the enrichment in the non-responders. And we used this set to be able to see if we can predict the non-response. And this is just based on our own data alone. And what we see is that if you have a higher enhancer activity, you do see non-response in, let's say, 13 out of 14 samples or so. And that can be predictor of this enhancer high or low signature can be predictor for survival, overall survival or progression-free survival. And if we apply our data with the Kelly's lab data, there is, with a decent amount of AUC value, we can actually see that this replicates between the two data sets. So this suggests that because we now have a discovery cohort and a validation cohort, this suggests that this epigenomic biomarkers, epigenomic content could actually be predictive of the immune therapy response. And this could potentially be developed along with other features of immune therapy non-response, for example, low infiltrate in the non-responder tumors, higher activity of PTEN mutation, for example, or a specific microbiome content of non-responders. One could potentially think of a multi-omic sort of biomarker type of signature that could potentially be used for stratifying patients into non-responders or responders for PD-1 or CTLA-4 therapy. And then in our the next question, we ask if could we then specifically target the non-coding elements themselves? So could we target specific enhancers for therapy itself? And that hasn't been done at all in the field. So for that, we went back to our pan-cancer data, the first data that I showed you, which is on the CCLE cell lines. And CCLE cell lines and the Sanger cell lines are really are very powerful in that there is a bunch of drug response data sets available as well for them. So we made use of the CTRB version to 545 compounds, and we overlap the non-response and response with the enrichment of enhancers or not. Then we made use of the Avana data, the CRISPR screening data, to identify genes that are essentially essential for in the non-responders or the responders lines. And then we overlaid the enhancer, the first set of analysis with the second, to ask in which setting the enhancer mediated gene activation likely leads to a non-response. And we found a bunch of correlations in this particular analysis, and that's shown here on the table. And breast cancer was one of the top one. And this is just shown here. What you would see is there is a bunch of genes for which we found a bunch of enhancers that can actually predict response to several inhibitors. So I'm going to show you one or two examples here because I find it really interesting. And here it is a region where you have enrichment of this enhancers around in the resistant lines, but not in the sensitive lines. And this marks the gene called FOXA1, and this is essentially a selumetinib plus a BRAF inhibitor combo. So, and you see that FOXA1 is highly expressed, and as well as the base factor is higher in the resistant lines, which suggests that this enhancer potentially targets FOXA1, which could be targeted for therapy in this particular, in the resistant lines for this particular combination of MEK plus BRAF. This can also be applied to this can also tell you the reverse setting, for example, here, where this set of super enhancer was actually activated in the sensitive lines for various EGFR and other inhibitors. And this essentially targets a bunch of different genes on chromosome 17, and there's another super enhancer that actually targets a bunch of genes on chromosome 8. And what you see is the dependency of all 8 genes to be higher in the sensitive lines compared to the resistant in chromosome 17 as well as in chromosome 18. What that means is potentially there is this super enhancer essentially activates all of these genes. And in fact, I don't have the data here, but we have evidence to show that all of these genes and this enhancer lie within a TED domain or within particular domain and talks. So this enhancer talks to each of these genes. And therefore, rather than targeting each of these genes, if you were to target this enhancer itself, you might get a much better response. So this would be a one agent rather than 8 different agents. So that shows you the power of targeting a non-coding element on its own. So enhancers can be potential drug therapeutic targets over, and in fact that could leverage the recent progress in RNA-based therapeutics. And enhancers make these things, make these RNAs, not all of them, but a lot of them would make these enhancer RNAs called enhancer RNAs. And essentially these enhancer RNAs then could be targeted with these RNA-based therapeutics. So this is something to do in future where we could potentially target specific enhancer elements based on ASOs or other types of agents that could target enhancer. So we'll work towards that as we go forward in the future. Finally, the genome, as I alluded to earlier, the genome is not linear. And in fact, uh, as I said earlier, that 2 meters of genome is actually packaged within the 10 micrometer space. So there is a specific set of folding events that occur that make sure that this huge compaction can occur within a tiny space. And indeed, it's been found now that this is quite regulatory in nature. And there are these loops that you can see here, for example. And these loops form something called TED domains, which are here. And these TED domains eventually form something called compartments. And compartments form something called chromosomal territories. So now one could think, and this is the nature of that. So for example, in the insulated neighborhood, this is the minimal unit. You have two CTSCF sites which loop in and bring an enhancer together with the gene. And these loops could actually contain many genes and many enhancers within one loop and or within one TED domain. So the scintillating idea that we are now trying to address is, could we cleave off one of these TED domains or one of these loop domains to be able to make cells better responders? And this can be done best in the T cells or in the adaptive or in the NK cells, where adaptive T cell or adaptive NK cell therapies could be used, that are being actively now being investigated for clinical use. So what one could think about is, and this is what we're trying to do, is to ask if in a terminally exhausted T cell, could you reverse the phenotype and make them more proliferative and tumor killing by simply taking out some of these domains or one or two of these domains. And you can imagine a future where you would take out either the endogenous T cells or the TILs from the patient. You could modify one of these domains and then put it back to the patients for a CAR T therapy or a CAR NK type of therapy. And the advantage of targeting domains is that it contains a regulatory unit, right? And a TED domain would be the basic fundamental unit of gene regulation. So you would have multiple enhancers and multiple TED domain, multiple genes together nested in one, and you can target the CTCF sites and cleave off specific ones and get a phenotype, desired phenotype. So with that, I'll thank people who have done the work. A lot of the work and a couple of these stories were led by Mahinur, and in equal collaboration with Chris, Jonathan, Elias, and Isis led the colon data, and Tommy helped with the immune therapy. This is an active collaboration with the immune therapy data is in active collaboration with Manolis and Genevieve. We're thankful to many people within MD Anderson, the Integromics book group for the colon cancer project, Jen and others, melanoma group for the samples, and obviously the sequencing course and the various other courses, and we are funded by various different agencies. So with that, I'll thank my lab. Here we're trying to kill cancer, and take any questions.
Thank you very much. Thank you, Dr. Rai, for this interesting presentation and really exciting work. We have a question from the audience, and the question is the following. It is interesting that in the PCA, the different tumor types don't necessarily cluster separately. Can you please comment on what shared factors, microenvironmental or otherwise, are responsible for these shared enhanced patterns?
So I would like to point out that these are cancer cell lines. So these are relatively homogeneous population. We don't have any tumor microenvironment component here. So we believe that these are purely driven by their similar epigenetic makeup rather than by the histological. And that's why we are interested in targeting them based on epigenetic therapy. I hope that answers the question. But I'm happy to be reached later on via email to answer more.
Thank you very much. Do we have any other questions? I think Dr. Kalin had a question, but I can't hear him. Let's see. If there are no, I think Dr. Kalin is trying to speak, but I can't hear him. So if there are no other, I'm sure he can catch up with you later. And again, thank you everyone for your attendance. Thank you very much to the organizer team, Alison Gart and Elizabeth Kent, for helping with this series. We will have another exciting presentation tomorrow from Dr. Phil Jones, and then starting next week and through the summer, the program will continue with once a week, Friday at 10 a.m. So thank you everyone, and I hope to see you tomorrow.
Thank you.
Thank you.